Hello and welcome to AP Psychology. My name is Mrs. Seguin and today we're going to be covering the brain part 2. So this part of the brain is going to focus on neuroplasticity. So our objectives for the day, we're going to discuss the role of neuroplasticity in traumatic brain injury, and then we're going to identify the contributions of key researchers in the study of neuroplasticity. Alright, the first objective. We're going to discuss the role of neuroplasticity in traumatic brain injury. Neuro, of course, refers to the brain. Plasticity is a new word, though. This is the brain's ability to change, especially during childhood, by reorganizing after damage or by building new pathways based on experience. So basically, it is the brain's ability to grow and change and to develop over time and to recover from damage. The key question here is, to what extent can a damaged brain reorganize itself? And then what is neurogenesis? So can we recover fully from a damaged brain? That's the real question here. So we have two main methods of brain damage. The first one is severed neurons. Now on a macro level, for example, if you were to sever your spinal cord, it's probably not going to regenerate. You're going to be permanently paralyzed. But on a micro level here, each neuron, if you sever a neuron, if you destroy a neuron, it's not going to regenerate most of the time. It's very difficult for neurons to regenerate. The second method of brain damage is that certain brain functions are pre-assigned to certain spots on the brain. And so if you damage a specific spot on the brain, you can damage your ability to do something. So basically what will end up happening, for example, a newborn who suffers damage to the temporal lobe facial recognition areas is never really able to recognize faces for the rest of their lives. So it's a more localized version of brain damage. The good news is that some of the brain's neural tissue can reorganize in response to damage. Under the surface of our awareness, the brain is constantly changing. It builds new pathways as it adjusts to little mishaps and new experiences, like falling and hitting your head. That causes minor levels of brain damage. This is not concussion level stuff, but it is minor levels of brain damage, and the brain has to go, oh, what do I do? And fix itself, right? Okay, so how do we repair the brain? One of the really cool ways that we do this is what we call constraint-induced therapy. So basically, this is someone who has probably damaged one of those parts of the brain or has damaged certain neurons, right, that control a limb. So they restrain the fully functioning limb. So let's say we have a bad arm and a good arm, right? A bad arm and a good arm. And they restrain that fully functioning limb, the good arm. And they force the patient to use the bad arm to do things, right, in their normal everyday lives. Eventually, this reprograms the brain to figure out how to make that arm function. How? Functions that would normally take place in the damaged part of the brain that is affecting your ability to use the limb now move to other regions of the brain. We call this reassignment. And reassignment is when brain functions migrate to other brain regions because the region of origin is damaged. So the part of the brain that something would normally happen from, what would normally control a certain movement, gets damaged. The brain can move the function to another part of the brain so that the body can continue to function. Examples of this: blindness or deafness makes unused areas of the brain available for other uses. If you're blind, you're not going to be able to see, so that visual cortex in your brain is unused. When blind people read Braille using the tips of their fingers, the brain area dedicated to the fingers expands into the visual cortex, because essentially when a blind person is reading Braille, they are seeing the reading, right? Another really cool example of this is that some deaf people have enhanced peripheral vision because in the absence of sound stimulation, because they can't hear, the temporal lobe area dedicated to hearing is now available for use by the visual system associated with sign language. So they basically hear through their eyes, essentially, if that makes any sense. If a second or third example here, if the left hemisphere area dedicated to language is disrupted by, say, a tumor, the right hemisphere can compensate. So the brain learns to adapt. It reassigns portions of the brain to different functions. The visual cortex should not be able to process bumps on a page, right? The temporal lobe should not be able to pick up sign language as hearing. Okay. One of the biggest ways we repair the brain, and it doesn't happen a lot, but it's a really cool way, is neurogenesis, and this is the formation of new neurons. Now, the cells in the body, other than neurons, are regenerating all the time, but your brain cells, they don't regenerate. If you kill one, it's dead. So the brain prefers to repair itself by reorganizing the existing tissue, so by using reassignment, right? But sometimes it becomes necessary to produce new brain cells. So we have these things called stem cells. Basically, stem cells are unspecialized cells. These are cells that don't have a job yet. Essentially what this does is you can take baby neurons from deep, deep, deep in the brain, and they can migrate elsewhere in the brain to form connections with other neurons to rebuild that neural connection, that neural pathway. So stem cells found in the human embryo, basically the idea here, this is a theory, right? That we are doing further research on this, is can we use stem cells to repair the brain by taking them from the human embryo and implanting them in the brain and assigning them to being neural cells, right? So that's something that's in further research. It's really interesting though. Okay, objective number 2. We're going to identify the contributions of key researchers to the study of neuroplasticity. Our biggest researcher here is Michael Gazzaniga. So he studied with Roger Sperry, who we talked about in part 1, and he studied those split brain epileptic patients who were missing their corpus callosum. So they had the 2 hemispheres of the brain, but they were divided instead of having the corpus callosum connecting them. Information about the right hemisphere, information from the right hemisphere about the left visual field and somatosensory signals from the left side of the body is no longer transmitted to the left hemisphere. So in other words, the body's not communicating properly. All right. Gazzaniga developed one of the biggest theories of consciousness. He basically developed this idea that your brain is constantly telling stories and that it builds stories out of the things that you perceive and that you sense, right? So that everything makes sense in your head. So I'm going to let you watch about a 3-minute video clip with him telling you these things, and then we'll come back.
Some people, for instance, love the metaphors that come out of, say, literature and the arts, and think that you want to leave those whole and untouched by an analysis as to why we may like those metaphors, or why those metaphors seem to help our thinking, or any of a number of questions. And there's a sense that if someone goes in and assesses that situation and gives a biologic dimension to why we do like those things, that somehow it's all diminished by that. And that's just not true. In my early part of my career, and continues, I studied patients who had their 2 brains disconnected. What we were responsible for was working out the functions of each hemisphere. You could work with them independent of the other influencing it. So you kind of got to study it alone. And over many, many years, the basic finding is, of course, that one brain didn't know what the other one was doing. Such people, that the information doesn't transfer between the brains. We found out that in the left brain, there's a special system that seems to always want to explain actions and moods that we have after they occurred. So we would put a question to the right non-speaking hemisphere, and it, in effect, would direct the left hand to do something. And so the patient would do that. And then we would simply say to the patient, well, why did you do that? The patient would make up a story that would explain why their hand had done one thing and why the other hand had done another thing, and wove a tale that made coherent, as it were, the behaviors that are coming from all these separate brain areas. The behavior comes out, and then there's this little narrator up there that turns it into a story that makes us feel coherent and unified. Turns out it's a thing in the left hemisphere that does this. And we called it the interpreter. And it's a very powerful force in the human condition, and it's always trying to figure out and seek explanations for our behavior. And that's what it does. I think the human as a storytelling animal, as some people put it, is because this system is continually trying to keep the story coherent, and even though these actions may be coming from processors going on outside of initially of conscious awareness, an action is produced, and then want to explain that action is to be part of your coherence and your story, your narrative. Why does the human always seem to like fiction? What is that? Could it be, as some people have suggested, could it be that prepares us for unexpected things that happen in our life because we've already thought about them in our fantasy world and or read about them in a fictitious setting and saw how those characters acted, and so then when we're confronted with it, we're ready. We've sort of lived through that movie, as it were. So why do we like that stuff? Well, maybe that's a reason why we like it. And to think of all of those things seems to me just to make it all richer, a richer experience.
All right, so in this lesson, you learned about the role of neuroplasticity in traumatic brain injury and about the contributions of key researchers. We talked about Michael Gazzaniga to the study of neuroplasticity. All right, I will see you in class tomorrow. Bye.